Hi Jessica, how are you?
I'm good, thank you. Thanks, Bel.
Good, good. How's your lockdown been?
Well, it's more than a year now, especially working in the university. I think we are very much really obeying what the government said. So I've been working from home for a whole year now.
So, have you always wanted to be a scientist when you were a child?
So I grew up in Hong Kong. And then I also did my undergraduate and also postgraduate study in Hong Kong before coming to the UK. And so I think Hong Kong being a more of a commercial business center, I think most of the people that probably wouldn't think of becoming a scientist. At least it wasn't my childhood dream. But as soon as I went to the university, then my view changed. I remember the first year in my undergraduate study, I did biochemistry, so it is not exactly neuroscience.
And how did you progress to your position at Spinal Research?
So what I did in my PhD was also the similar molecule that I'm looking at now. So it's a class of molecule, it's a carbohydrate actually, so it is a sugar. So I was working on this particular sugar molecule, but it is mostly related to the development of the nervous system. So towards the end of my PhD, then I thought I'm quite bored about development. I want something different, but at the same time, I'm still very interested in the sugar molecule itself. So then I thought, okay, maybe I can actually look at other system or maybe some disease that is related to that particular molecule. And then I was also thinking that I would like to go outside Hong Kong to get some external experience, foreign experience. It is always good to do it in research as well. I want to go to countries where there's a long history, and also they are having good scientific excellence as well. So I came to the UK in 2005, did my postdoc in Cambridge together with James Fawcett at the Brain Repair Centre.
Oh, wow.
Yeah, and that is how I progressed from Cambridge as a postdoctoral scientist, then a senior research associate, and then moved to Leeds about 6 years ago to start my own group here.
What is your area of research at the moment?
So the sugar is called chondroitin sulfate proteoglycan. I'll keep it simple, a sugar molecule. Yeah. So a sugar molecule in the central nervous system. And this particular molecule actually is also present in our joints as well, normally. And then what is interesting is after injury, this sugar will be expressed by the cells around the injury site. So there will be a high upregulation of this molecule. So you have a lot around in the surrounding. And it has been proven that they are actually inhibitory to regeneration. What we are doing in the lab in the last, I would say 10, 15 years is to look at whether we can find a tool which we can downregulate or remove this sugar from the lesion site and then enhance regeneration. So that was one of the aims we set out around 10, 15 years ago. And then later on, we also realized that this sugar is also present on the surface of neuron. I use one analogy all the time to explain this code. It's basically like the plastic sheath on an electrical cable. So in order to make connection, you need to remove the plastic, and then you make the wire connection, and then you need to reseal it with the plastic again. This sugar layer on the surface of neuron is doing exactly the same. So when it is there, it is actually limiting more synapses to form. And synapses are the main connections between neurons. That is how we think, and then we move our body to different muscle. So if you remove this layer, then there are more opportunities for the synapses to talk to each other. And then after they make the synapses, after they start talking, then you just put this plastic layer or this sugar layer back to the neuron, and then they will fire more efficiently. So that's the two phenomena we saw. One is around the lesion site, one is on the surface of neuron. And what we are aiming in the lab is to find a tool which we can remove this biochemically, ideally to be something that can be administered easily into the body. But that is what we've been working on.
And how do you think that this research can help someone like me?
Yeah, so these two events, So one is, as I said, so let's say this is the spinal cord. Yeah, you have the lesion sites there, you have the glycan upregulated here. On the other hand, on the surface of neuron, you have this plastic insulation layer, or the sugar insulation layer. And these are actually two events for regeneration. If we remove the glycans here, we can encourage regeneration. So we can encourage neurons going through the sites. If we remove the insulation layer on the surface of neuron, we can make more synapses. And this is what we call as plasticity. So regeneration itself, it takes a long time. So if you remove the sugar right after injury, you can enhance the regeneration at the beginning. We call it acute or subacute period. So you can enhance the regeneration. But the growth of neurons still, on the other hand, is very slow. So what we are aiming is that we can also enhance local connections. So after they pass the lesion sites, can they actually connect locally and then use whatever that has been spared, not injured during the initial injury, to make the functional connection. And by enhancing plasticity, what we expect is that it is not just helping the acute situation, it will also help the chronic patients in regeneration. I mean, obviously, we are not having a magic bullet there. It's not a magic potion that will regain all the function, but certainly I think partial recovery of function is achievable in both acute and chronic population pool.
So do you think that the length or the time after injury can affect the outcomes of the research?
From our understanding of the biological system now, it should not be affected by the time. So mainly there are different reasons for that. I think one is that there's one paper published about 3 years ago, I think, by a group in America. So they have looked at, again, in animal model, in that case, and looking at animals which has been injured for 1.5 years already. And in that case, even just one single treatment downregulating this sugar is sufficient to recover the function, or is a respiratory function in that case. So the animal actually recovered the function within 1 week after the treatment.
Oh, wow.
So it is 1.5 years after injury, one single dose, and then after a week, then the animal actually recovered the respiratory function. So, and that was with a tool which is a direct enzyme injection into the area. But nonetheless, we know that if we can remove the glycan in the area, we can enhance regeneration and we can enhance local connection. And that seems to be sufficient even in a chronic model.
And historically, what has the progress been like in treatment of spinal cord injuries, and what is the current state of play, do you think?
There are lots of different, how would I call it, stabilizing treatments to patients after spinal cord injury, but none of these treatments are actually helping the regeneration or itself. So that is the scenario we are sitting right now. So can we actually enhance regeneration and also maximize the plasticities? Rehabilitation itself is also promoting plasticity. But obviously, we also see that rehabilitation itself is not sufficient to enhance functional recovery. So can we open up the surface of neuron by removing the plastic insulation or this glycan coat, and therefore maximize the possibility for making more synapses? That is what we call plasticity. And then for the recovery of function. And that is what we are aiming. So a completely new line of treatment theory. But hoping to combine this together with other existing treatments and promote functional recovery in the spinal cord injured population.
Obviously, there's a lot of supporters of spinal research and people like me determined to kind of help in any way we can. And how do you think the fundraising can help the work that you're doing?
So thank you again, Bel, in the first place, because I think your support to the community is to the research funding. It is really important. So I think so there are a few different aspects here. One is the patient's number is relatively low in the spinal cord injury community compared to other diseases. So, and because of that, a lot of the big funders would not be taking spinal cord injury as a distinct support area per se. So I think increasing the awareness both to the funders and also to the society that spinal cord injury is a long process. Patients are generally having a long lifespan, relatively normal life, but on the other hand, they are of course limited to the wheelchair or bear with different types of disability. So the cost, the care cost or the healthcare cost for the patients is really huge. So dependent on, of course, also the level of the injury as well. So can we actually increase the awareness in the society of how much we are actually spending in the population in supporting particularly the young people who have sustained spinal cord injury, and also because the central nervous system injury in terms of research is also taking a lot longer time to other areas of science. So we really need to have a lot of funding in order to support the work that we are doing. The patients community has been really supportive, and what we need to deliver is really to go out to the society and make them aware that this is actually a very severe impact to patients' life, and we need to put more resources into it.
Thank you so much, Jessica. It's been really nice to speak to you.
You're very welcome.
It's amazing what you're doing as well. Thank you.